Hello, everybody. Welcome to today's talk in our Evolution and Ecology seminar series. Today, we are very excited to have with us Dr. Siobhán O'Brien. Siobhán is a tenure-track fellow at the University of Liverpool. She obtained her undergraduate degree from Trinity College in Dublin, and her master's and PhD from the University of Exeter. After her PhD, she was a postdoc at the University of Exeter, the University of York, then the ETH in Zurich, and finally at the Wissenschaftskolleg in Berlin, before taking up her current post at the University of Liverpool. In 2018, Siobhán won the prestigious John Maynard Smith Prize for Evolutionary Biology. Siobhán researches how microbial communities evolve, particularly how interactions between species influence evolutionary outcomes such as adaptation to changing environments. To do so, she uses experimental evolution in complex communities in various environments. She continues to pursue these questions in a recently received BBSRC Discovery Fellowship, during which she explores how species interactions shape adaptive evolution in soil microbial communities. Today, Siobhán will talk to us about ecology and evolution of microbial social lives. Siobhán has pre-recorded the talk, which is the manifestation of a compromise over parent-offspring conflict over who gets this time slot. However, she is live with us in the background, and we will have a live Q&A session with Siobhán after her talk. So please do post your questions in the designated Slack channel. And now, without any further delay, here's Siobhán O'Brien.
Great, so I'd first like to start by thanking the organisers for giving me the opportunity to speak at this seminar series. I think it's so great that something really positive has actually come out of COVID times, really. This seminar series has given, it's brought evolutionary biology to such a wider variety of people that normally would be exposed to it. So thank you so much for doing that. I'd also like to thank Andreas as well, because this is actually the first talk I have given since being on maternity leave. So I've been on mat leave for the last year, and I'm back in work full time now, about 6 weeks. So he's really given me a good kick to sort of get the brain cells firing again. So hopefully they're alive enough to speak to you about my science today. Okay, so since finishing my PhD in 2015, I have worked in about 5 different institutions. So I moved from Exeter up to York, over to Switzerland, to Berlin, and of course now I'm really happy to be in a tenure track position at Liverpool and finally be able to get my teeth sort of stuck into something more long term. Why do I say this? I say it because it's quite difficult, obviously, to sort of knit together a really cohesive narrative about your science when you're doing these sort of short-term postdoc positions on lots of different systems. So, there's been a great benefit in getting to live lots of different places and working with amazing people on lots of different systems, but it is hard to knit things together. And so if you see me trying to make a very tentative link between different topics, this is why. Okay, so I am really fascinated by social interactions from bacteria to humans. And I'm really interested in understanding how social interactions could matter for things like virulence, cleaning up heavy metals for the environment, antibiotic resistance, and so on. And bacteria are really fantastic model systems to ask these kinds of questions. So, for instance, species of Saccharomyces cerevisiae, they group together to form flocks when the environment is kind of, when there's like harsh conditions in the environment. And this is a cooperative behavior. And we also have things like division of labor in microbial colonies, where colonies at the external surface actually act as sort of moisteners to allow the internal cells to sort of shunt their way forward to allow them to explore new environments. And so I use microbes to ask questions about social interactions in lots of different contexts. So ranging from very simple fundamental questions in test tubes consisting of a couple of species or one species, all the way to more applied contexts, for instance, in the cystic fibrosis lung, gut microbiome and soil microbiome. And I first paradoxically, I suppose, got interested in microbes when I was doing my master's project on white-browed sparrow weavers in the Kalahari. And I remember sitting in the field site and just reading some papers about social evolution. And I kind of got down a rabbit hole about social evolution theory and ended up here at the social lives of microbes. And this is actually a really seminal paper in microbiology by Stu Weston and Ashley Griffin. And I was just really, really blown away at the fact that you could answer very similar questions as I was trying to answer in birds, but on bacteria in the lab. I thought this was just amazing. And I'd never even stepped foot inside a lab at this point. So this was it really blew me away. And so I ended up doing my PhD on siderophore production, probably because of reading that paper. Angus Buckling was actually one of the authors on that paper, who I ended up doing my PhD with. So that was really great. I worked on siderophore production. So bacteria often find themselves in iron-limited environments, and they need iron to grow and survive. And so to overcome this, they produce iron-chelating siderophores. And the siderophores are able to bind iron, so here, and then they can use the siderophores to sort of shunt the iron into the cell. And then the siderophore is released back into the environment where it can be reused. Of course, anyone with the correct siderophore receptor can really benefit from siderophores. It doesn't just have to be the producer. So you often get sort of non-producers evolving quite quickly that don't produce siderophores, but that can benefit from the siderophores produced by others. Siderophores are quite costly. And so if you grow a non-producer just in isolation in iron-limited media, its fitness tends to be much lower than the producer. So here in this graph on the left-hand side bar, the fitness is less than 1 of the non-producer. And this means that it's not growing as well as a producer would be in this environment because it can't access iron. But when you put the non-producer in the presence of a producer, so in co-culture, actually the non-producer is now gaining a huge fitness advantage because it can use the siderophores produced by the producing strain, but is not paying the cost of producing any itself. So siderophores carry a cost. And because of this, if you evolve a single siderophore producing clone in iron-limited media, this is over about 25 days, siderophore cheats or non-producers can evolve really, really rapidly. So after 25 days, you can see at least 4 replicate populations where cheats are reaching over 80% of the population. So they're really, really high frequencies. And so the 2 questions or 2 topics I'm going to talk to you about today are, firstly, whether or not cooperators can actually adapt to having cheats in their population. And also, subsequently, does it actually matter? Why might this matter? And secondly, I'm going to talk about cooperation and exploitation in a community-wide public good. So this is a cooperative trait that's not specific to a certain species, but can potentially benefit lots of different microbes in the community. Okay, so to first ask whether or not cooperators can actually adapt to having cheats in their population. So in the animal kingdom, there's lots of ways we know cooperation can be maintained in societies. So for instance, through kin selection, so directing the benefits of cooperation to kin, and policing and punishment in human societies, and through things like reciprocity, for example. But actually, in microbes, we see very similar features. So for instance, in D. discoideum, antagonistic pleiotropy can be a form of punishment. So cheats avoid differentiating into the non-reproductive stalk cells here, But these cheats are subsequently, they can be excluded from these reproductive spores. So cheats are almost punished for cheating in this scenario. And there's similar examples in the Saccharomyces example I spoke about earlier, and with division of labour. However, in microbes, many cooperative traits are actually diffuse public goods, such as siderophores. So it's less clear of how sort of cooperators can mitigate the adverse effects of cheating when you can't directly identify cheats in this context, so when things are more diffuse. So to try and investigate whether or not this could occur, we set up this really simple experiment where we evolved populations comprising of either 100% cooperators or 10% cooperators and 90% cheats. So in this second treatment with cheats, there was really strong selection for cooperators to try and mitigate the adverse effects of cheating. We evolved these populations for about 200 generations. And after this time, we were able to pull out only cooperators, so they were tagged, and look at the fitness response to selection. So had cooperators actually been able to adapt to the presence of cheats, and also do some phenotypic assays on cooperators. So for instance, measuring siderophore production. So in order to first test the fitness response to selection, we competed all of our evolved populations against our ancestral population. And on the y-axis here, I'm showing you the fitness of evolved populations relative to the ancestor. So when this value is 1, both evolved and ancestral populations have equal fitness. When this is less than 1, our ancestral populations are doing better than evolved. And when this is greater than 1, our evolved populations are doing better than the ancestor. And so when this is greater than 1, there's some evidence for adaptation. And what you can see is that at least in 3 of these bars, we find that the relative fitness of evolved populations is actually less than 1. So it doesn't look like there's much adaptation to the environment. But because we were specifically interested in adaptation to cheats rather than the environment per se, we actually did this computation in the presence and absence of cheats. So the darker colours are in the presence of cheats and the lighter colours in the absence of cheats. And we can see that here in this bar, in this bar, this bar on the furthest right, the relative fitness of evolved populations is higher than 1. So there is some evidence for adaptation. And this is when this is the populations that had evolved with cheats. They're fitter than the ancestor, but only in the presence of cheats. So it seems like they have specifically evolved to the presence of cheats. But the question is, how have they done this? Well, to try and answer this question, we first of all measured siderophore production here on the right-hand side. And on the left-hand side, we measured pyovirgin production, so the principal siderophore of Pseudomonas aeruginosa. This is in Pseudomonas aeruginosa, by the way. You can tell I am a zoologist at heart rather than a pure microbiologist. So what did we find? We found that populations that had evolved in the presence of cheats actually produced less siderophore than those that had evolved without cheats and the ancestor. And this is actually quite counterintuitive. So I think we initially hypothesized that actually cheat evolved cooperators should probably have overcompensated for the fact that there's lots of cheats around. So we expected them to be producing more siderophore production overall to try and compensate for this high cheating load. But of course, this is the opposite. And it was also really puzzling because we didn't find any cost to this behavior. All of our populations really had the same growth rate, irrespective of the amount of siderophores they were producing. So it does seem like they're finding some way of accessing iron that isn't siderophores, because there doesn't seem to be a cost to producing siderophore production in an iron-limited environment. Now, luckily, I'm probably quite a messy person in the lab and tend to leave my plates on benches far longer than I should. But I started to notice that we're getting a lot of these small colony variants on the cheat-adapted population plates compared to our cooperator-only plates. And we were actually finding these morphotypes on about 5 out of 6 cheat adapted populations, reaching quite high frequencies in many of them, almost up to fixation. And when you grow these novel morphotypes in just normal lab media, they're this really characteristic blue sort of green color. And this color tends to be associated with the toxin piocyanin. So piocyanin is a toxin probably most well known in the context of infections because it damages host cells. It can make patients quite sick. And lo and behold, we measured the piocyanin production for our cheat-evolved populations and our populations evolving without cheats, and found that cheat-evolved populations were producing more piocyanin than those that had evolved without cheats. We also sequenced these novel morphotypes and found large sort of large genomic deletions consisting of about 19 genes. And within this region, there was mutations in repressor and promoter regions. So this suggests why we get a sort of upregulation of piocyanin and downregulation of siderophore production. And we never found these deletions in smooth morphotypes, which was interesting. So piocyanin is a really, common toxin, but actually it's more than just a toxin. So piocyanin itself is a really powerful iron reducing agent. It can turn iron from the unavailable Fe3+ form, which requires siderophores, into a more available Fe2+ form, which can simply just diffuse into cells. And so we hypothesized that actually maybe populations were reducing their siderophore production, upregulating piocyanin production as an alternative way to access iron. And so to test this, we added exogenous piocyanin into populations of just cooperating wild-type Pseudomonas aeruginosa and to cheating populations. And we measured the relative fitness of each strain. So this is the fitness of each strain relative to itself in the absence of any piocyanin. And what we found was that if you add exogenous piocyanin to cheat populations here in blue, the cheats are gaining a massive advantage, whereas we don't see the same level of advantage in cooperating populations. And this is because cheats are able to use piocyanin to access iron, whereas the wild type the cooperators don't need to do that because they're already producing siderophores. So altogether, I think this is really good evidence that the cooperators are adapting to cheats by downregulating their own siderophore production and instead using piocyanin to access iron. Okay, so this was all work that I sort of did towards the end of my PhD. And afterwards, I moved up to York to work in Mike Brockhurst's lab. And there I was looking at sort of the evolution of virulence in the cystic fibrosis lung, specifically in Pseudomonas aeruginosa. And here I was met with piocyanin again. So on these plates, I've plated isolates from the cystic fibrosis lung. And you'll probably notice, first of all, that lots of these plates have this real characteristic blue-green sort of piocyanin color. But you'll also probably notice that it's very varied, okay? So some plates have lots of piocyanin and others have very, very little. However, we still don't really understand what drives this variation. So why do some patients harbor so many piocyanin producers while some don't? And why does the proportion of piocyanin production change within a patient through time? This is the variation in piocyanin production that we measured in cystic fibrosis patients. So on the x-axis is a snapshot of Pseudomonas aeruginosa isolates in 3 different patients. And so there's 40 isolates per patient at a given time. And we measured piocyanin production for each isolate. And all I want you to take from this is the fact that in 2 of these patients, there's so much variation in piocyanin production. So we've got colonies that are almost 3-fold higher than some other isolates that aren't producing anything. And then we have this other patient here that's basically composed of colonies that aren't producing much piocyanin at all. So much variation there. So what is driving this variation? Well, we know that piocyanin is generally a toxin that harms host cells. So we know that the host response is probably important. But actually, I've just shown you evidence that within species interactions, specifically competition for iron, can also drive piocyanin production. And piocyanin is also a really important antimicrobial, so it can reduce the diversity of species in the environment and can also sort of trigger inflammatory responses in other species. And these inflammatory responses can actually turn species that are classically commensal and kind of harmless into potentially more virulent ones. So it's clear that social interactions are really, really important, but it's less clear about how sort of this occurs. And so to try and investigate this, I had two really, really great master's students in the lab last year, Axel and Maddie, and they've started to do some really nice preliminary experiments to try and tackle some of these questions. And so they set up this really simple experiment where they grew up Pseudomonas aeruginosa, a wild type normal piocyanin producer here in the blue bars, but also a piocyanin negative mutant, so here in red. And they found that when they grow these species just in isolation, there's not really much in terms of the difference in growth rate. If anything, the piocyanin negative mutant is actually growing a little bit better. And potentially this is because there's some cost associated with piocyanin when it's not needed. But they also grew these different strains in combination with species that we commonly find in the cystic fibrosis lung. And in this case, they found that piocyanin producers are actually getting quite a sizable advantage in the presence of these other species. And we don't see the same effect in piocyanin non-producers. So this suggests that piocyanin is quite an important determinant of the nature of interactions between species in the lung. So they then went further and asked, okay, well, what's piocyanin actually doing to these other CF lung species? So to try and get at this, they grew up Pseudomonas, our Pseudomonas strains. So we had our piocyanin producer in blue, our piocyanin negative mutant in green, and then just normal LB medium in red. So they grew up all these strains, and this time they actually filtered the cultures to remove Pseudomonas aeruginosa. So they were just left with the supernatant, so anything that Pseudomonas produced. And this time they grew each of these species in the presence of this Pseudomonas supernatant or an LB control. And they didn't find much of an effect of the supernatant on Raleigh dentacaryosa or Staph epidermidis, but they did find quite a strong effect on Staph aureus. So if you grow Staph aureus up in normal LB and Pseudomonas aeruginosa piocyanin mutant supernatant, so no piocyanin, there's not really much of a difference in terms of growth rate. But if you grow it in the presence of a piocyanin producing supernatant, the growth of staph is really dramatically reduced. So again, this shows that piocyanin is really shaping the interactions between these two species. So it seems really clear that piocyanin is important, at least for Pseudomonas aeruginosa and staph interactions. And so to try and explore this further, they set up a really nice coevolution experiment. So in this experiment, they had two treatments. The first treatment consisted of Staph aureus, evolved alongside a Pseudomonas aeruginosa piocyanin producer. And in the second treatment, they had staph evolved alongside a non-producing mutant. So the only difference between the two treatments was whether or not Pseudomonas was actually producing piocyanin. And at the end of the experiment, they wanted to assess the cell densities through time, so to see how Pseudomonas was affected by whether or not it could produce piocyanin, and to see how staph was affected by whether or not Pseudomonas was actually producing piocyanin. They wanted to assess the fitness response to selection, so how had or if adaptation had occurred in the two strains, and the genetics response to selection, so how has adaptation occurred. Unfortunately, this experiment was started early 2021 And they were just about to start on these second two tasks when COVID sort of closed the labs down. And so we have lovely data for cell densities through time in Pseudomonas, but unfortunately we haven't managed to collect this data yet. And Axel and Maddie have since gone on to much greater things. They finished their masters, so it's really down to me to get my ass back in the lab, to be honest, and hopefully finish this project. But until then, I'm going to show you the cell densities through time, because I do think it's kind of indicative of some interesting relationships between the two species. So what we find here is that this is just for Pseudomonas aeruginosa, by the way. We don't have the staff data just yet. But for Pseudomonas aeruginosa, the blue line, this is the piocyanin producer. And you can see after about 6 days, there is a benefit of producing piocyanin. So the populations that are producing piocyanin tend to be overall slightly growing slightly better than those that are not producing piocyanin. We also see some interesting things going on though. So there's weird overlap where at the start of the experiment, we're seeing that it almost looks like, again, piocyanin is carrying the cost, which is weird because it is in the presence of Staph aureus. But we also know that Staph and Pseudomonas interactions are quite complex. Staph can, the Pseudomonas can sometimes trigger a sort of switch in lifestyle with Staph. It can trigger a switch from it being aerobic to anaerobic fermentation. And this can alter the virulence of Staph. And so there's probably lots of really interesting things going on here, which we'll be able to find out, I suppose, when we do some sequencing and some more experiments. But I'm really excited by this data and I'm looking forward to getting more information. And so to quickly summarize this part of the talk, we've shown that cooperators can adapt to siderophore cheats by reducing their own siderophore output. To compensate, they're producing more piocyanin, which, while it's an important toxin, it can also liberate iron. And this switch can potentially influence community dynamics, especially in the cystic fibrosis lung, potentially, but also with regards to Pseudomonas and Staph aureus interactions. Okay, so on to the second part of my talk now, and we're going to switch gears entirely and look at heavy metal contamination. So heavy metal contamination is a really big problem worldwide. It's caused by things like fertilizer applications, pesticide use, but mostly mining activities. And because of the negative effects it can have on human health, there's a real global drive towards identifying contaminated sites and trying to clean them up. But to be honest, there's been really mixed effects. So in Europe, we've identified about 2.5 million contaminated sites, but actually only successfully remediated about 15% of them. So luckily for us, microbes interact with heavy metals big time. They've lots of ways to keep heavy metals out of their cells. And some of these methods can be really beneficial for us because they also reduce the toxicity of heavy metals in the environment. So one example is the production of siderophores. Now, unlike iron, in the context of heavy metals, siderophores can't take them up. So while free copper can simply diffuse into cells, once the copper is bound to a siderophore, it can no longer enter bacterial cells. So this keeps the bacteria happy because the toxicity is reduced by siderophores. It keeps their neighbors happy because the toxicity is reduced for them as well. And it keeps us happy and the environment happy because the heavy metal content in the environment is reduced. However, we've already shown that siderophores are vulnerable to exploitation in the context of iron, and in the context of heavy metals, there's no difference. So in the lab, we evolved a single siderophore producing clone of Pseudomonas aeruginosa in the presence of copper and in just a copper-free control. And after about 200 generations of evolution, we're getting about 35% cheats evolving de novo. So within species exploitation seems to be really common, at least in lab experiments. we also looked at potential for exploitation between species. So we showed that siderophores produced by one species can potentially help another. So again, if you grow a producer, here are the black bars, a non-producing strain, just in copper, in this case is Pseudomonas fluorescens, you can see that the producing strain grows better than the non-producer, presumably because it's producing siderophores. But if you grow them up again in the presence of these exogenous siderophores produced by different species, this cost of not producing siderophore is ameliorated. And so the benefit of cheating becomes greater. Basically, they're able to use siderophores produced by other species. So does this fact that siderophore non-producers evolve so rapidly explain why using siderophores or siderophore producing species has such mixed results in nature? Well, no, probably not, because the experiments I have just discussed are in really simple lab environments, and in soil things can be pretty different. So in soil, we've got things like spatial structure, for instance, and spatial structure can keep producers and their products together. And this means that cheats are less able to access the benefits of cooperation. Soil also contains a really complex natural microbial community. And so cheats can not only benefit from their conspecifics, but potentially could benefit from siderophores produced by anyone in the soil. So maybe the opportunities for cheating are really exaggerated in soil. We don't know. So we set up a really simple experiment to try and investigate this. So here we grew up a single clone of Pseudomonas fluorescens producing strain, so here in red, and a non-producing strain in white. We grew them up in monoculture conditions, so they're grown alone, and together in co-culture conditions. And this time, instead of lab media, we grew our strains up in soil. So in this case, it was sterilized, autoclaved compost soil. And then to test the effect of the community, we re-added in a soil wash containing the compost community, so here in blue. And crucially, we treated this community as a black box, so we didn't really know what was in there. All we knew is that it allowed interactions to take place, not just within species, but between different species. We did know there were some siderophore producers in there. We did this experiment in copper enriched and control soils, and we left them all for about 5 days, after which we counted colony forming units on plates, so we assessed cell densities. So first of all, is there evidence for within species exploitation, so between different strains of Pseudomonas fluorescens? So in this graph, I'm showing you the fitness of a siderophore non-producing strain relative to the producing strain in the same environment. So when this value is equal to 1, there's no difference in fitness between the producing and the non-producing strains. When this is less than 1, we see that the non-producing strain is doing worse than the producing strain. And when it's greater than 1, the non-producing strain is outcompeting the producing strain. And there's lots and lots of different comparisons we can make here. But what I want to show you is basically these two bars that are highlighted in black. So this bar here is showing you the fitness of the non-producer grown alone in soil in copper. So as you'd expect, the non-producer is not really doing very well here. And that's because it's in a toxic environment, but it's not producing siderophores. So it's obviously not very fit. But actually, if all you do is just add in a producing strain, which is this bar, you can see that the non-producers are gaining quite a sizable fitness advantage here. So this is strong evidence for within species exploitation in soil. What about between species? Can non-producers benefit from the community? So again, focus on this non-producing growth rate here. If all we do this time is add in the natural community, we also see a fitness increase. So again, this is the non-producing strain, grown alone in copper, very sick, not growing well. Add in the natural microbial community and bam, the fitness is increased. And interestingly, when you add in both, so when you add in your producing strain, if you add in your producing strain and your community, you're getting the biggest fitness increase, which is this bar here that I'm pointing at. So this seems like quite strong evidence for between species exploitation in soil. And this means that as the amount of heavy metals gets bigger in soils, we'd actually expect more siderophore cheating as the cost of cooperation becomes bigger. And this means that you get less siderophore production where it's most needed. And so this is kind of the exact opposite of what we want from a bioremediation perspective. If we're adding in siderophores or siderophore producing bacteria into soil to try and clean up the soil, we want more siderophore production, not less, when there's more contamination around. And so to check whether or not this is the case, we very excitedly went into the wild for about 90 minutes. So to the bemusement of our field ecologists colleagues, we went to an old mining site in Cornwall. And so this is the Paradise Valley. It was heavily mined in the 18th and 19th century. And it's not worked anymore, but has left a kind of a rich legacy of mining waste. And we were able to collect samples across a gradient of heavy metals. So we collected 94 samples, ranging from highly contaminated areas around sort of slag heaps, all the way to regenerated areas. And among lots of other things, we measured the proportion of siderophore producers per sample, as well as heavy metal content. And there's lots of different correlations we can make with this data, but the one I want to draw your attention to is in this black box here. And this is showing that as you get more heavy metals in the environment, we find more siderophore producers. Now, this is clearly a very tentative, positive interaction, but the important thing is that it's not negative. So we don't see this signature of cheating where we would get more we don't see evidence of cheating where we would get more or fewer producers when there's more heavy metals around. We find the opposite. We find more siderophore producers when there's lots of heavy metals around, which doesn't sound like there's much cheating going on. But of course, these are all correlations, and so we don't really know what's driving this correlation. And so to try and see what was going on, we did some experimental evolution back in the lab. So again, doing these experiments in soil. This time we did some evolution. So we basically had a focal species of Pseudomonas fluorescens, just a normal producer. And we either grew that alone or we embedded it within a natural soil community, or we just grew the community by itself. And we set up this all up in controlled populations or controlled environment with no copper and in copper-rich environments. And so with this experiment, we were able to sort of pick apart what was happening at the community level and within an individual species. And this work was done with Elsa Hesse down in Exeter, and she's a postdoc now with Angus, and we had some really great fun doing this. And Elsa and Angus are continuing to do some great work in this area, so keep your eyes peeled for their stuff. Okay, so let's look at community siderophore production first. Here I'm showing you the mean siderophore production over the course of the 6-week experiment. And the white dots depict the treatments in the absence of copper, and the black dots are depicting the treatment in the presence of copper. So you could probably immediately see that in the presence of copper, we're getting higher siderophore production than in the absence of copper. And this is driven by species sorting. So we did some sequencing of clones isolated from our community treatment, and we found that in copper evolved communities, we were getting sort of sorting towards high siderophore producing species such as Vivario vax and Microbacterium. And communities that had evolved in copper-rich environments had then displayed increased metal tolerance at the end of the experiment. You can see from the bottom graph here, compared to communities that had evolved in the absence of copper. And so all of this suggests that cheating is not really very important. We find more siderophore producers in toxic copper because species sorting allows selection for high-producing species. So what about within our individual focal species, Pseudomonas fluorescens? Well, in this case, we're looking at mean siderophore production here on the y-axis over the course of 6 weeks again. But here we're looking at it in the absence of the community and the presence of the community on the right panel. And so in this case, we can see that in the presence of copper at the end of the experiment, Pseudomonas fluorescens is producing lower amounts of siderophore, which is the opposite of what we find at the community level. And so this does suggest that cheating is important, at least within this species. But the other thing that could be driving this is, of course, just losing siderophore production because it's not needed. So a loss due to disuse sort of scenario. And the way of teasing this apart, so cheating versus loss due to disuse, is to test whether something is negatively frequency dependent. So cheating is always negatively frequency dependent, right? Because you need to have some cooperators in the environment to get a benefit of cheating. So the more rare a cheat is, the bigger the benefit it can get from its cooperators that are also there. If something is just lost due to disuse, it's usually not negatively frequency dependent because it's not needed irrespective of how common it is in a population. And so we tested this in soil by competing siderophore producers and non-producers in control and copper conditions from rare and from common. And on the y-axis here, we have the selection coefficient. So this is how fit a given strain is in competition with another strain. And so the higher the selection coefficient, the fitter the strain. And you can see that the fittest strain we have in our whole experiment here is the non-producing strain in light grey that's competed from rare. So our non-producing strain is getting the best fitness advantage when it's rare in the population. We don't see this same fitness advantage in a copper or in a control environment. We only see it in copper. And I think this is really strong evidence overall that this is genuine cheating that's driving the loss of siderophore production. Okay, so to really summarize sort of 5 years of work in a really simple graphic, this is really what we found. So at the level of the community, species sorting is selecting for high siderophore producers. While sort of at the level of individual species, we have cheating playing a role in reducing the amount of siderophores being produced. And ultimately, this means that cooperation sort of stabilizes at intermediate levels. You don't want to be too high a producer because you'll be exploited by non-producers, but you don't want to be too low either because then you'll be outcompeted when species sorting occurs in response to copper. And so, as I mentioned, Elsa and Angus have continued to sort of explore this really nicely, and I'll try and explain it now. And so on the left-hand side, this is basically showing a histogram of siderophore production for the copper evolved communities and control communities. So the copper evolved communities are these red humps. And so in the copper evolved communities, we lose low producing species because they are not selected for during species sorting. But we also lose high producing species here because they are exploited. And they also found that looking at six different species they found in the community, they found that species that generally produce quite a lot of siderophore in a control environment downregulate the siderophore production in copper, and vice versa for species that produce little siderophore in a control environment, they upregulate their production in copper. So there seems to be this real drive towards producing an intermediate level of siderophore production. Okay, so I hope I've managed to convince you that social interactions are just really, really amazing. And they're able to sort of predict lots of really interesting things about ecosystem level processes, so such as detoxification, virulence, and also, you know, other things like antibiotic resistance and so on. And so if you can really understand what's driving the emergence of cheats, being able to manipulate social interactions could be really powerful in helping us to enhance natural processes, such as for biotechnology. And with that, I'll say thank you so much for listening. This work was done by lots and lots of amazing people, and I had so many great collaborators over the years. But I'd like to thank the main ones. So Angus and Elsa for all of the soil stuff, Mike for the piocyanin production in natural CF isolates, and Maddie and Axel, who were wonderful to work with and great master's students at Liverpool. I'd also like to thank all of my funding bodies, especially BBSRC for a discovery fellowship. And with that, I'd love to take any questions that you might have. Thank you very much.
All right, thanks very much for this. As questions start to come in, maybe I'll start with some of my own. Is there a possibility for any strains to use both siderophores and piocyanin, or is that always kind of, it's either one or the other because doing both is too costly or doesn't make sense? Sorry if this doesn't make sense because I'm not a microbial.
Yeah, no, definitely. I would say both are important at the same time. Piocyanin, yeah, it's important for accessing iron potentially, but it has lots and lots of other functions. So as I mentioned, it is important in sort of responding to the host immune system. It's an antimicrobial, you know, it's upregulated in response to lots of other things. And its function as an iron reducing agent is generally stems from its role as a sort of a redox active molecule that sort of causes horrible oxidative stress and so on, and it's linked to lots of other sort of virulence factors. So I mean, yeah, the question remains as to how much is piocyanin upregulated in response to iron versus all this other stuff, and then its ability to access iron is kind of a byproduct potentially. But it is likely that they're somehow related. So the more piocyanin that's produced, potentially less siderophores might need to be produced because there could be more iron in the environment in that case. But they could certainly be producing both, yeah, depending on the environment.
I don't know if it's a follow-up. I think it's a follow-up from that. Is iron really important? Iron seems to be quite important in the lab environment, but equally it seems quite important within the lungs of CF patients. Are these similar environments, whereas soils might be quite different in the availability of nutrients?
Yeah, sure. So in CF, I think the common belief is that iron is actually quite available on the large scale. So if you measure the amount of iron in CF, it tends to be quite large. However, I'm not sure, and I'm sure there's many people that know more about this than I do, but I'm not sure if they actually measure available versus unavailable iron. So while there might be a lot of iron present, we don't know how accessible it is for bacteria. And then the other thing is that obviously we're talking about bacterial interactions on kind of a micro scale. So it's really hard for us to really determine whether or not iron is actually available on that micro scale or not, obviously. And there's also, so other people have also done great experiments looking at, for instance, evidence for social cheating in the CF lung. So Sandra Anderson had a lovely paper out in PNAS a few years ago. And basically there's quite good evidence that cheating is important in the lung. And so that suggests that actually iron is limited as well, if you look at it that way. In terms of soil, there's so many different soils with lots of different pHs, and pHs are a really important driver of iron availability, obviously. So Again, I think it's quite context dependent. And that's one of the kind of caveats, I suppose, of what we're doing, is that we're obviously picking these soils and these composts that sort of suit our questions. And the question still remains as to, okay, well, that's great, it suits our questions, but what happens in the real world? And so what we're trying to do is kind of bridge the gap, I suppose, between the lab media, right, and going out into the environment, is to try and get somewhere in the middle. And yeah, it doesn't answer sort of questions about what's happening in soil more generally, but it provides a good model system, I think, to answer questions about things like communities, how communities might be important, and spatial structure and things like that, things that are just extra on top of sort of the lab environment.
Yeah, well, certainly you've got to start somewhere, right? You can't just go and pick any old soil. So sure, it makes sense to start with something that suits your lab needs as well. So I'll go over to some questions that are coming in from the audience. I think the first one is kind of a clarification. So could you explain again your 2017 result about piocyanin? Is piocyanin benefiting cheaters? And then the person goes on to say, Dandekar and Greenberg have shown that piocyanin reduces cheater invasion.
Oh yeah, so I think they're talking about that. So I think that was quorum sensing cheats in that paper, rather than siderophore cheats per se. But it is possible that piocyanin could also prevent the emergence of cheats in the same way of siderophore cheats, because if piocyanin is acting to increase the amount of iron in the environment, then the benefit of cheating is reduced. There's no point in being a cheat because there's no cost to producing siderophores. And with regards to just clarifying the experiment, I suppose the experiment that I've just shown was adding piocyanin into just populations of cheats and showing that they can benefit from having more iron. So a little bit different, but certainly the impact of piocyanin on siderophore cheating could be the same as the paper, on quorum sensing.
All right, thanks. The next question is, it actually references a paper again. People are very specific here. Harris in 2013. The title is Dynamic Social Behavior in a Bacterium, Pseudomonas aeruginosa partially compensates for siderophore loss to cheats. Sorry. They found that cooperators in the presence of cheaters produce more siderophore relative to when only cooperators are present, which is a result quite different from yours. And could you comment on that?
Yeah, sure. So I know Freya really well, and yeah, I do know this paper, and there was a few papers that have tried to look at this sort of adaptation to cheating in siderophore production, and I think in this paper they didn't actually measure, so there was no selection for high fitness or productivity, which we had in our experiment. So any sort of adaptation to cheats couldn't come at the cost of population productivity and fitness. And that was kind of an important distinction, which it can actually ultimately alter evolutionary trajectories quite substantially if there's no fitness consequences in terms of population densities. So slightly different.
So more generally then, I don't know if again this makes sense. In the general context, do you think doing these fitness assays and kind of in isolation, which I assume is still kind of the gold standard or the default for most people working in microbiology, do you think that is often a good indicator of these fitness things, or should we move generally more towards testing the fitness in these different environments that also involve species interactions?
Yeah, I suppose it depends on the experiment that you've done. So in our case, we tend to test the fitness effect. What we're testing there is the evolutionary response in our evolution experiment. So what we're testing is, have the strains become adapted to whatever we've specifically selected for in that experiment. And the power of that sort of fitness assay is that you can tease apart stuff like, has it adapted to the lab? Probably, or in my case it didn't, weirdly, but usually it does adapt to the lab. versus, okay, well, it's lab adapted, but has it adapted to some other factor that you've included, such as cheat. So you can really pinpoint what's allowed them to increase in fitness. And then normally you can sequence them, right? And show that there's a mutation that's responsible for driving this. But again, that's sort of a real simple experiment. And yeah, for sure. I mean, I think it's really great if we can make things more complex and add in more species and get whole communities involved. You know, I think understanding microbiome function, for instance, is so important, but we're so far from doing it because it's just so complicated. So I'm all for that. But I also think sort of fundamental experimental evolution is just really fun and and can tell you really powerful things as well. So even though it might not be applied to a specific thing, it's just really interesting, isn't it, to sort of understand how evolution happens. So you know, big fan.
Yeah, I agree. I'm always really jealous of these you know, seemingly easy, if you don't do it, comparisons with, you know, just compare it against the ancestor and stuff like that. That in I don't know, in animals, otherwise it's quite hard to just keep your ancestor somewhere and then do direct competition. So it is really cool stuff. I really like it. Okay, so there is another question that's coming. Is there a high turnover rate of non-cheating to cheating populations? As in, does it happen quite quickly?
Yeah. So you do. I think what they mean is yeah. So do you get cheating evolving quite fast? Yeah, you do. So it really depends on what you're selecting for or how you're selecting for cheats, I suppose. So for instance, in the detoxification context, so when we were looking at cheats in response to copper, cheats were evolving so fast and to reaching such high densities, there was really strong selection for cheating. With iron limitation, even though it's kind of the known way of looking for siderophore cheats, it's slightly lower, but we're still getting, like, you know, over a matter of 20 days or so, cheats can reach really sizable frequencies in a population, so 80-90%. So, you know, as a human, yeah, they evolve super fast. But obviously, it depends on how strong selection you're making for cheating, but you can make the selection quite strong. And I actually forgot what I was going to say. Oh yeah, so the stronger, so basically the more iron is limited, or the more copper there is around, the more costly it is for an individual cell to produce siderophores, because they have to produce so much of them. And so it's under those conditions that you get more cheats evolving really quickly, because it's just so costly to produce siderophores. So weirdly, it's kind of the opposite to what you'd expect. When you need cooperators around, you're actually more likely to get cheats. So it's a positive.
Sorry. Is it typical for cheats to be kind of loss of function mutants essentially? And if so, can they re-evolve the cooperator trait?
They're usually like a point mutation. So they usually evolve, I think that's why they evolve so fast. It's just a simple point mutation. And I'm going to be so embarrassed now, because I know Rolf Kummerly did this experiment to look at whether or not cheats can re-evolve cooperation. And I've read the paper ages ago. Can't remember what he found, but look him up. So it was Rolf Kummerly's lab in Zurich did this exact experiment. I'm so sorry, Rolf. Yeah.
So, but in the piocyanin, it was large deletions, right? But
right. So sorry. So
then.
Yeah, so the point mutations are the siderophore production or producers. In piocyanin, we used it was a piocyanin mutant that somebody made. So it was isogenic from the wild type, but it had, in this case, it was actually phenazine. So it's kind of an operon that encodes piocyanin. So it was like, I think 4 or 5 genes deleted in that operon. But in terms of piocyanin negative mutants, I'm not sure. I think they can be coded by lots of different things when you look at them in natural populations. So it's all sort of controlled by the quorum sensing system, which is just a web, and I'm not even going to attempt to try and explain now, but basically there's lots of ways you can get to piocyanin negative mutants. And a lot of the time, if the main sort of core sensing genes are mutated, you don't just get loss of piocyanin, you get loss of a whole like plethora of virulence factors. So they tend to kind of produce all or nothing. But in our experiments, the mutants that we used were just piocyanin mutants.
Cool. I think we're just about out of time, but thanks again very much for your talk and also for actually joining us for the Q&A. Brilliant. That leaves me to announce next week's speaker, who is Melissa Rowe from the Netherlands Institute of Ecology in Wageningen. The title of her talk will be Reproductive Microbiomes and the Functional Evolution of Avian Ejaculates. So please do join us again next Wednesday, same time, same place. Make sure you keep up to date with our schedule by following us on Twitter, Evo Eco Seminars, and or by joining our Slack channel. Thanks for watching.